Hello everyone. Today we are going to discuss about the particular disease that is called herpes disease. It is caused by herpes virus, and this topic belongs to the course MMB205, module number 3. Herpes virus is an enveloped DNA virus. It has spherical icosahedral symmetry, and the diameter is about 150 to 200 nanometer. So this particular virus has been regarded as the second in size only to pox viruses. If you look into the picture, you can see that this particular enveloped virus contains lipoprotein. The yellow region or the orange region is the lipoprotein envelope. And from this lipoprotein envelope, you can see some projection that is green in color. These projections are glycoprotein spikes. So this envelope is nothing but the lipid bilayer. They are deriving this lipid bilayer from the nuclear membrane of the host cell and containing these viral glycoproteins. Below this envelope layer, you can see this purple color layer. This purple color layer, which surrounds the capsid and the DNA core, is known as tegument. So it's a protein containing region. And just beneath the tegument, these green bubbles, they are the capsomer containing the capsid, means they're actually preparing the capsid for the virus. And inside the capsid, they have a double-stranded linear DNA. This particular DNA can encode more than 35 proteins. So this is the overall structure of herpes virus. Next slide is about the classification of human herpes viruses. So about 8 human herpes virus species are known to date. They are herpes simplex virus type 1, HSV1, or can be called as human herpes virus 1, herpes simplex virus type 2, or human herpes virus type 2, then varicella zoster virus, which is HHV3, Epstein-Barr virus, cytomegalovirus, human herpes virus type 6, 7, and 8. HHV8 is also known as Kaposi sarcoma virus. So this is all about the classification of human herpes virus species. So 8 species are there that can cause different type of diseases that are going to be discussed through this lecture by herpes viruses. So classification is not only done by the first slide, what I have said, that 8 classifications are there, but classification can also be done on the basis of biologic characteristics. So on that basis, herpes viruses can be classified into 3 different classes, 3 different groups. The first group is known as alpha herpes virini, beta herpes virini, and gamma herpes virini. Herpes simplex virus group HSV1 and HSV2 falls into the first class, that is alpha herpes virini. Cytomegalovirus group pauses in the second class rather, the beta herpes virini, and lymphoproliferative group belongs to the gamma herpes virini. So the right hand side picture will tell you how they are classed on the basis of the biological characteristics. Now, herpes simplex virus, type 1 and type 2, they are actually distinguished by two main criteria. What are these two criteria? This is the first criteria is antigenicity, the glycoproteins they have. And the second one is the location of lesions. So they are animal viruses that create disease to the human. So where, in what location actually they infect? On that basis, herpes simplex virus can be classed into two different types. The first class, that is HSV1, here they are actually creating lesions or infection primarily in adults, and here the lesions they are creating above the waist. What are the clinical manifestations there? So HSV1 can create acute gingivostomatitis, recurrent herpes labialis, labialis, that is cold sore, or keratoconjunctivitis, or simply it can be called as keratitis, and encephalitis. HSV2, that is actually creating lesions or infections below the waist, they are responsible for herpes genitalis or genital herpes, neonatal encephalitis, and other forms of neonatal herpes, aseptic meningitis. But whatever be the clinical manifestations, whatever be the position or location of lesions, both these two type of viruses use human as a host. Today, through this lecture, we will cover the infection caused by HSV1. So these are the clinical manifestations caused by herpes simplex virus. So variety of clinical manifestations can occur due to the infection of HSV, and those are listed here: acute gingivostomatitis, herpes labialis, ocular herpes, herpes genitalis, other forms of cutaneous herpes, meningitis, encephalitis, neonatal herpes, like that. Now, after entering into the human cell, how this particular type of virus replicate? We have already discussed that this particular virus is an enveloped virus. They contain double-stranded linear DNA. So after entering into the host cell, the virus will uncoat its capsid and release the DNA into the cytoplasm of the host cell. Then the DNA migrates to the nucleus, and into nucleus or within the nucleus, they transcribe to form mRNA by using the host cell RNA polymerase. They don't have RNA polymerase. They are actually using the host cell RNA polymerase, that is DNA dependent RNA polymerase. Then after synthesis of mRNA or after transcription, mRNA will come out again to the cytoplasm and will go for protein synthesis. And when they have synthesized protein, then within the nucleus, the viral DNA will replicate using the machinery of the genomic DNA, and they will come out, assemble with the proteins, and will produce a new virus. So this picture will clearly indicates that how HSV replicate. So number one, you can see that fusion between the virus glycoprotein and human cell surface receptor took place. So after fusion, there will be internalization of the virus particle. So the virus particle will enter into the cytoplasm via endocytosis, and then after the entry, they will uncoat and will release DNA into the cytoplasm of the host cell. So then some carrier protein will guide the viral DNA to the nucleus membrane, and then they will enter through the nuclear pore, and after entering within the nucleus, they starts DNA replication as well as transcription. So by the process of transcription, they are actually preparing mRNA using the host cell RNA polymerase. So after formation of mRNA, the mRNA will come out again to the cytoplasm in association with endoplasmic reticulum, better to say in association with the ribosome. They form three different types of proteins: immediate early protein, and lead proteins. When the lead protein, they have synthesized the lead protein, then the lead protein will come within the nuclear environment, then association or assemble with the viral DNA, and then fuse with the nuclear membrane. Here, this particular type of virus is preparing their envelope not from the cell membrane but from the nuclear membrane. And after formation of envelope, they will again synthesize some lead protein, that is the glycoprotein, the spikes protein, and they will come out via endoplasmic reticulum and Golgi apparatus from the host cell. So this is a particular type of replication process you people we haven't discussed so far. Now, how they transmit? So we will focus here only HSV1 and HSV2. HSV1 is primarily transmitted in saliva, so oral, and HSV2 is transmitted by sexual contact. So oral genital sexual activity can transmit both HSV1 and HSV2. So you can find via oral genital sexual activity HSV1 infections of the genital and HSV2 lesions in the oral cavity. And here 10 to 20% of cases you can observe. So in case of HSV2 infections has markedly increased comparatively HSV1. So statistics have shown that HSV2 infections has markedly increased comparatively. So what are the clinical findings? So HSV1 can cause several forms of primary and recurrent diseases. What are the type of diseases? They can cause gingivostomatitis. The first picture you can see occurs primarily in children and is characterized by fever, irritability, vesicular lesions in the mouth. You can see white patches, vesicular lesions in the mouth. The primary disease is more severe and lasts longer than the recurrent one. Normally, these lesions heal spontaneously in 2 to 3 weeks. Many children have asymptomatic primary infections. The second type is herpes labialis. This is cold sores. Is a milder one, a recurrent form, characterized by crops of vesicles, usually at the mucocutaneous junction between the lips and the nose. Here in the second picture tells you what is actually herpes labialis. Recurrences frequently appear at the same site in case of herpes labialis. Then keratoconjunctivitis. characterized by corneal ulcer and lesions of the conjunctival epithelium. And in case of recurrence, can lead to scarring and blindness. The first picture. The second picture tells you about encephalitis. So necrotic lesion in one temporal lobe caused by HSV1. Here the symptoms are maybe fever, headache, vomiting, seizure, and altered mental status. The fifth one is herpetic white lobe. The first picture tells you about the disease. So pustular lesion of the skin of the finger or hand. It can occur in medical personnel as a result of contact with patient's lesion. And the second one is herpes gladiatorum. So wrestlers and others who have close body contact may have this particular type of disease. So vesicular lesion on the head and neck and trunk can be observed.